John, let me know when you want me to put the slides on.
Okay, Andrew, that's working fine.
Okay.
Thank you. Ron, I'm going to share a couple slides first.
Okay.
Okay, hello, welcome everybody. My name is John Aitchison, and I am the editor-in-chief of Molecular Biology of the Cell. And it's a pleasure today to host this webinar on really highlighting the special issue that MBoC has had ongoing for a little bit, forces on and within cells, with a keynote presentation by our ASCB President Rong Li. The goal of this webinar is to introduce you to some of the really fantastic science that's being done by some of our more junior people in MBoC, and to promote the global visibility of some of the really fantastic science, as well as the scientists ongoing, and through ASCB and molecular biology of the cell. The special topic has published over 110 research articles, and we encourage those who are interested in biophysics and understanding the forces on and within cells to contribute to this collection. The lead editors are all here: Valerie Weaver, Dennis Diescher, Alex Dunn, and Alpha Yap. And they will each introduce some of the papers that that have been selected for presentation in the webinar. Before we begin, the special, and I will ask each of them to introduce those speakers. And right now, I'm going to ask Alex to talk a little bit about this rally board. Alex, are you there?
Uh, I
Oh,
actually I think John, it's
Dennis
Dennis.
is
Yeah,
going to do it. I'm
yeah. If
sorry.
you're gonna,
I
yeah,
thought
yeah. No,
I saw it on my screen. Yeah.
that's okay. I'm the guilty one who sent this to you. So this was sent to me earlier today by ASCB, and really all the information I have is in front of you, and I think the important thing is. Watch your inbox at the very bottom. Watch your inbox for more details on this. So this is, really within ASCB as opposed to what John told you about MBoC. I hope we're collecting, all the email addresses for the participants, and if not, send us your email address. And as it says here, watch your inbox for more details about this.
Thank you,
That's it,
Dennis.
John. That's it, John. Yeah, thanks.
So I'm going to hand it over now to Valerie Weaver and to introduce our keynote speaker, uh, Dr. Lee.
Okay, so my name is Val Weaver. I'm one of the editors on this special issues, and it is my pleasure to actually introduce our keynote speaker today, Dr. Rong Li, who is basically currently our president of ASCB, which is fabulous, and basically is director of the Mechanobiology Institute in Singapore. And prior to there, she was a distinguished professor at Johns Hopkins. And Dr. Lee has a really distinguished career, and she's published amazing work. She's a top scientist, and I'm so looking forward to her presentation. She studies cellular dynamics and mechanics, and she focuses on understanding the morphogenesis, stress adaptation, and aging in eukaryotic cells. And today, her talk is going to be on dynamics and mechanics in development and aging of eukaryotic cells. And without further ado, I hand over the podium to my esteemed colleague, Dr. Lee.
Thank you, Valerie. Can you see my slides?
We can.
Yep.
Okay. Yep. Yeah. Good afternoon, everyone. It's really an honor to give this keynote at the webinar on forces on within and within cells, or within and on cells. I would like to really congratulate our lead editors for a fantastic collection. So, as Valerie has introduced, I'm currently the director of the Mechanobiology Institute on the campus of National University of Singapore. So I'm a cell biologist at heart, but I truly believe that mechanobiology is a frontier in cell biological research. So there are many definitions of mechanobiology, but we think of mechanobiology, really based on a very broad perspective. That is to uh, attempt to understand biological systems from the perspectives of physics and mechanics. But there are focuses, and one of the important focus is forces and how are forces produced and regulated to drive form and function, and how are mechanical inputs transduced and integrated with biochemical signals. What I also really love about mechanobiology is that it's a truly interdisciplinary field. It requires expertise from various fields in engineering, physics, biology, all come together and solve complex problems. And this is a community that we're trying to establish at MBI. And also, we really want to continue to build interdisciplinary community at ASCB, continue to expand our diversity and bring people together from various fields. So a current theme at my institute is mechanobiology of aging, because aging is a great risk factor for many wide variety of human diseases that we still do not have cure. We believe that mechanobiology holds the key for understanding aging and treating age-associated conditions, because mechanical stress are often cause of wear and tear on tissues and cells that lead to diseases. But at the same time, forces and mechanics can provide the signals as well as the means for repair and regeneration. So we're hoping that through our research, we can learn how to tune biological mechanics to minimize damage and to promote better repair and regeneration. So today I'm going to just give you a highlight of a line of work sort of along this theme. And that's addressing age-associated decline in female fertility. I'm very fascinated by the fact that the female reproductive system is the fastest aging system of the human body. So when a woman is only about late 30s or early 40s years of age, she's still a very young person, but her reproductive system has already declined tremendously. Her ability to have children has reduced. And that's associated with the decrease in both the number as well as the quality of the follicles that carry oocytes, which is a precursor cell that give rise to human gamete, female gamete egg. The cellular basis of follicle quality decline is associated with oocyte aging. Hallmarks of this are low rate of meiotic maturation and high frequency of producing aneuploid eggs. So here's a young mouse oocyte undergoing meiosis. You can see it occurs in an asymmetric manner and also segregates chromosomes properly. And here's a representative faulty meiosis of an aged mouse oocyte. You can see chromosome segregation is very sloppy, generating aneuploid eggs. Another hallmark of aged oocyte is metabolic dysfunction. And one of the current theories in the field is that dysfunctional metabolism or mitochondria can give rise to high level of ROS, which generate oxidative damage on a very important structure called cohesins. Cohesins are these ring-like structures that keep homologous chromosome and sister chromosome together. And during aging, when they're damaged, they're not being replenished. So that leads to premature chromosome separation and giving rise to chromosome missegregation during meiosis. This is a very compelling hypothesis, but we think it might be just the tip of the iceberg. So work in my lab and others in the field in the past decades have been studying asymmetric meiotic cell divisions during oocyte maturation and zygote formation. And here I just want to give you a summary of the dynamic and mechanical processes that are going on within the oocyte that underlying this process to give you an idea that how much dynamic and energetically expensive process that needs to happen in the right way in order for an oocyte to produce a healthy egg that can be fertilized and produce a healthy zygote. So the first step in this asymmetric meiotic division is movement of the spindle from the middle to center to a subcortical region in order to extrude a polar body. And our work have led to a model where this process is driven by actin polymerization nucleated by formin proteins on the surface of these ER vesicles surrounding the spindle. So here ER is a layer very close to spindle. And now actin polymerization is between the ER layer and another layer of organelle, mitochondria. And so our collaboration with Shang San's group have shown that this system of actin polymerization can spontaneously break symmetry and generate directional spindle motion. And then once the first meiosis finished, the second meiotic spindle assembles subcortically and must be positioned here stably until fertilization. And we have shown that this positioning and maintenance of spindle position is carried out by a treadmilling actin array nucleated by ARP2/3 complex at this polar cortex, generating a cytoplasmic flow that maintains spindle position through a hydrodynamic force. Then after fertilization, sperm entry, the egg finishes meiosis 2. And this involves not only chromosome segregation, but also rotation of the spindle about 90 degrees in order to extrude the second set of chromosomes. And so and we have shown that this involves two actin-based forces. One is orchestrated by ARP2/3 complex, and the other one is contractile force generated by myosin 2. And these forces are directed in opposite directions and generating a torque on the spindle to rotate the spindle. So the reason I wanted to go through this very quickly is that this leads to our current hypothesis that metabolic dysfunction of aged oocyte could potentially not only generating oxidative stress to impair, to damage structures such as cohesins, but also can lead to reduced ATP production and impair skeletal dynamics, leading to failures in meiotic divisions. So to test this hypothesis and also understanding the underlying mechanisms, in recent years, we've been attempting to study follicle aging, not just looking at oocytes, but also the surrounding somatic cell environment in this tissue unit called the follicle. So in the follicles, the oocyte is surrounded by these somatic cells called granulosa cells. These cells are super important for supporting the metabolism of oocyte through these projections called TZPs. So these are cytoskeletal-based projections with gap junctions at the end, allowing the granulosa cells to transport nutrients and energy precursors to the oocyte to support oocyte metabolism. And oocyte in turn provides growth factors to support granulosa cell proliferation and survivability. So to study this, a very talented former postdoc fellow, Haiyang Wang, who just recently started his own lab in China, set up a 3D follicle culture system where we can take secondary follicles and culture in this 3D hydrogel system for it to develop and mature. And so this system we show that can recapitulate the aging phenotypes such as slowed follicle growth and increased follicle atresia. One of the first things we notice is that in an aged follicle, such as the example here, while the oocyte undergo aging, here as shown by increased reactive oxygen species, the surrounding somatic cells also undergo aging. So if you look at it on an individual follicle basis, the ROS level in the oocyte and the granulosa cells are highly correlated. So this tells us that the somatic cells and the oocyte age hand in hand. So that generated a question for us, who's causing who to get old between the granulosa cells and the oocyte? So to ask that question, Haiyan set up this second system, which we call reconstituted chimeric follicles, where we can take an oocyte, for example, from an aged follicle and then insert it into a young follicle whose own oocyte has been removed. So here's a movie of this. So we are removing the oocyte of this follicle and then putting in the oocyte from another follicle. And so this generates reconstituted chimeric follicle, or RCF, that we can be cultured in the 3D system and to further develop. So what we found is in this particular experiment, so with this, we can do various combinations. Here, I just want to highlight a really exciting set of experiments where we put aged oocyte in the young follicle. We ask, can a young somatic cell environment make an aged oocyte young again? So here we found that the young granulosa cells can more vigorously extend TZPs towards the aged follicles compared to aged granulosa cells, and generating an enhanced TZP density compared to aged follicles or aged oocyte in the aged follicle. And this perhaps together with the fact that the young granulosa cells are more metabolically healthy leads to metabolic restoration of the aged oocyte, as shown by single oocyte metabolomic analysis in collaboration with Chester Drum's group at NUS. And this leads to enhanced ATP production as well as reduced intracellular ROS. We think that the consequence of that, as we observed, is the improved meiotic segregation, as shown by reduced aneuploidy and separation of premature separation of sister chromatids in the mature eggs. And this is associated with, surprisingly, better preservation of cohesins on the meiotic chromosomes, and as well as decreased level of frequency of chromosome segregation errors, as we can observe through live imaging. And more excitingly, this leads to enhanced developmental potential of the aged oocyte. So we show that after this last developmental process in the young follicle, the aged oocyte have restored meiotic maturation, improved embryo formation, and very incredible, very excitingly, we show that when we implanted these fertilized two-cell embryos from these rejuvenated aged oocyte, they give rise to live birth 4 times higher than aged oocyte grown in aged follicles. So this is we think it's a very useful and powerful system for further understanding aging, as well as the interaction between somatic cells and the oocyte during these aging and rejuvenation. But we also get really excited about the potential to use this to improve reproductive health. So this is a recent development. Instead of using follicles, we can now take isolated granulosa cells and put them together with oocyte in a specially designed 3D system. Within days, these cells can self-organize into a fully functional follicle. And these artificial follicles, as we call them, have really almost identical developmental potential as the natural follicles. So now we can dream about what we call a cell-based therapy to enhance assisted reproductive technology such as IVF. So we envision that we can acquire oocyte from from older patients who have difficulty to get pregnant. We can surround these oocytes with young granulosa cells, which we're now working to engineer, and as well as engineered extracellular matrix with optimal biochemical and biomechanical properties. So we think of this as a five-star anti-aging spa for the old eggs. So after in this spa, the eggs will be healthy and rejuvenated, and then better give rise to healthy births. So this is the end of my talk, and acknowledge our collaborators and funding. Finally, I want to give a shout to Cell Bio 2026 in December in San Diego. We've just pretty much finalized a really exciting program with keynotes, symposia, mini symposia, and many different type of presentations and workshops, and also a brand new app, and also an app that can facilitate interactions and networking. So I hope to see all of you in San Diego in December. Thank you.
What a wonderful talk, Ron. Thank you.
Would you like me to stop sharing?
Sure, that would be fine. I think there might be time for a couple questions. Do you think
Okay.
we could host a couple questions, Valerie?
There's a question already in the chat. Alpha Yap asked a question. Is there a role, Lee, for fluid pressures in the follicle lumen that influence oocyte health or follicle cell function?
Yeah, so that's a great question. And so the fluid in the lumen occurs towards the very last stage of follicle development in the antral stage. So some of the process that we're aiming to support in a healthy environment occurs even before that. But fluid pressure likely to play a very important role in the ovulation process, where the oocyte matures and get ejected out of the follicle. And this is something that my colleague here at Mechanobiology Institute, Joe Chen, is actively studying. It's a really fascinating process. So, but it's not really well understood. Thank you.
May I ask a very different question, Ron?
Yeah.
What would be the regulatory challenges of this in vitro support of oocytes?
Regulatory, you mean not you mean human regulatory
Yes.
challenges?
Yes.
We are exploring that. But I think the fact that these therapeutic cells that we're generating, they do all their job in vitro outside of the human body. So after they've done their job, the egg that is ovulated will also be stripped off any remaining somatic cell, before their after fertilization and implantation. So none of these cells will go into the patient bodies. And we also show that there's no DNA transfer or mitochondrial transfer between the granulosa cells and the oocytes. So hopefully, these facts make regulatory hurdles less, a bit more easier.
Great.
Yeah.
Fantastic.
Thank you.
And promising, of course. A great example of how we go from basic cell biology to some real important translational applications.
As a basic biologist, cell biologist, when we try to solve these, look into these real world problems, I'm actually more excited about what won't work. And that presents, new questions and gaps in our understanding. So I also welcome that as well. Of course, we hope to make this work.
We have another question in here that says, I'm not familiar with the IVF protocols, but assuming the couple uses their own oocytes and sperm, do you think the metabolic dysregulation also impacts on cohesion in sperm chromosomes?
Probably not in the same way. So sperm and oocytes are very different in that oocytes are generated in full. All of the oocytes are produced actually during a late embryonic development. So, and these cohesins are deposited during that time, and they're not being replenished for years throughout the reproductive aging. Whereas sperms, are generated throughout the adult life and undergo, constant turnover. So I think male fertility has different types of problems. And I think there's really emerging research in this area. And I'm not an expert in that, but I think my impression is that mutations for older fathers could be more of a problem than the inability than failed meiosis. But I acknowledge I'm not an expert in that area. Yeah.
So, if there's no other questions, I'm going to pass the baton to the next person in line.
The next person in line happens to be me. And I just want to mention that, first of all, thank you, Dr. Li, for, really exciting talk. And over the next 35 to 40 minutes, we're going to have short talks from the speakers that you see on your screen now and summarizing recent papers published as part of the special issue. And I will now pass it over to Dennis to introduce our first speaker.
That's right. I'll introduce Chunzi Liu, currently at UC Berkeley. I believe she did the work that you'll hear about while at Stanford, and the title of the paper that appeared that she'll describe is delamination of epithelia induced by air-liquid interfaces. Please, Chunzi.
Thank you for the nice introduction. I'll share my screen and just let me know if it's working all right.
Looking great.
Oh, great. Cool. Thank you for the introduction, Professor Dister. And I just love how Professor Li's talk framed how mechanical force is manifested at the interface between follicle cells and oocyte. And I want to take on that thread and introduce one force that is almost always left off the map when we're thinking about mechanotransduction. So we've cataloged many of the forces as cellular interfaces. We have cell-cell adhesion, cell-substrate adhesion, or actomyosin contractility. But there is one force present at every single interface that we almost always overlook in a cellular context, which is the interfacial tension. Interfacial tension describes the energetic cost of increasing surface area at the boundary between two fluid phases. And you know this force intuitively. It's why two touching soap bubbles share a flat interface. And when one of the fluid phases is air, we call it the surface tension. In a cellular context, interfacial tension is typically dismissed as too small to matter. However, if we do a back of the envelope estimation, it will put the force, so capital force is the force generated by interfacial tension. If we calculate this at a two-cell interface, this will put us around 10 to the negative 7 Newton, roughly similar to the force that E-cadherin-mediated adhesion would generate per cell. And if that's not convincing enough, think about the following scenarios. At immune synapses, the adhesion is sparse, or at air-liquid interfaces, for example, an ocular surface, this force doesn't just participate, it actually dominates the cellular response. And that's exactly what we're trying to show in this paper. Here we used ocular epithelial cells cultured on collagen-coated cover slips. To create an air-liquid interface, we inverted the slides, used buoyancy to press an air bubble gently against the epithelial layers, and then tilt the chamber. As the bubble tried to escape under buoyancy, it will exert a force against the epithelial cells. And here's the video that we took from this device that we made in lab. Here the camera is rotating with the chamber, so it is easier to observe the cellular sheet. The angle on the upper left corner indicates the increasing force or the increasing angle. So here you can see that the cell layer is actually delaminating from the substrate, which is purely driven by the surface tension at this air-liquid interface. And towards the end of the video here, you can see that this cell layer fully recovered back onto the substrate. So that observation immediately raised the question, can we actually measure this force? Now, measuring solid surface tension has historically been very hard. Most methods only give relative values, such as contact angle measurements demonstrated by Barua and Parent et al. In 2017, where they measured the contact angle at a cell doublet interface and report a relative surface tension. In the paper we published, I developed a non-invasive assay to measure the cellular interfacial tension based on Scholl's method from 1977. So here, by measuring the cell contact angle against a series of calibrated fluid phase, we can extract the absolute cell surface tension. So here on this plot, the slope of these two lines help us calculate the surface tension. What we found is that the two ocular epithelial cell lines we cultured have measurably different surface tension, which in the context of the eye actually helps explains why the tear film stays so stably anchored onto the ocular surface instead of just evaporating away or dripping by the gravity. So finally, we asked how interfacial tension interacts with other forces on the cell surface. Here we used enzymatic and small molecule perturbations to systematically vary cell-cell adhesion, cell-substrate adhesion, and cortical tension. The result is a force balance picture that predicts three distinctive outcomes: a stable adhesion phase where the cells remain static, a delamination phase, which we have seen before, and a rupture phase in which the cells will dissociate from each other within the cell layer. And here, as you can see through the three videos, we observe all the three phases exactly as predicted. The take-home message here is that interfacial tension is a real, measurable, and sometimes dominant force at cellular interfaces, while it has been hidden in plain sight. So anywhere cells meet air, this force is in play, and that includes epithelial cells in the lung, gut, oral mucosa, as well as cell cultures under the airlifting condition. I'm also actively exploring how effective interfacial tension shapes immune response during this macrophage phagocytosis process. With that, I want to thank my PhD mentors, Jared Fuller and Alex Down. As Professor Dister mentioned, I'm currently a postdoc in UC Berkeley, studying how mucin-like glycoproteins modulate immune responses to parasites and bacteria. Happy to connect and answer any questions, and my email is on the screen.
Okay, thanks. If there's time, we'll take questions later. So I'll just pass it to Alpha Yap, who will introduce our next speaker. Alpha.
Let me
Thanks,
just add,
Dennis.
before you begin, Alpha, let me just add, if you've got questions for Shunzi or the next speakers, you can put them in the chat like Alpha and I have, and we'll get to them during the panel discussion. Thank you.
Thanks, John. And so it's my pleasure to introduce our next speaker, who is Andrew Stevens, who comes from the University of Massachusetts Amherst. Andrew is going to represent his lab's work, I believe, on nuclear mechanics and nuclear damage. So let me hand it all over to Andrew.
Can you see my screen okay?
All good.
All right, great. So I want to thank the presenters for giving me a chance to talk to you today about our story about how DNA damage causes ATM-dependent heterochromatin loss, leading to nuclear softening, loving, and rupture. This work is done by an undergrad in the lab at the time, Neviat, who has now moved on to graduate school. Sorry. So we're going to jump away from sort of tissues and large setups, and we're going to move straight into the cell, and we're going to be looking at the cell nucleus. And so in human disease, cell nuclei show abnormal nuclear shape and mechanics, and that's across the human disease spectrum. We look at a specific type of perturbation called nuclear blebbing. In a battery of publications we've had in the last year, we've clearly outlined that it's herniation of the nucleus away from the nuclear body. You can see here, and it has decreased DNA density, which is its main hallmark. And you should care about nuclear blebs because when the nucleus forms a bleb, it ruptures, as you can see here, through loss of NLS-GFP. And us and many in the field have shown that this causes cellular dysfunction, including DNA damage, altered transcription, disrupted cell cycle control, and inflammation signaling that likely drives the disease state. So we wanted to ask the question, what if these dysfunctions fed back and caused more nuclear blebbing and rupture? And we specifically focused on, would DNA damage increase nuclear blebbing and rupture? And so we treated cells with DNA damage agents, cisplatin, CSP, which causes DNA adducts, or bleomycin, which is a radiomimetic that causes DNA double-strand breaks. And through IF, we can see increases in DNA damage. And we also saw, along with increased DNA damage, we saw increases in nuclear blebbing and rupture. In any one of these panels, you can see a normally shaped nucleus and interphase forming a bleb, and then rupturing, shown as loss of NLS-GFP. So what is the mechanism behind loss of nuclear shape? Well, nuclear mechanics is constantly resisting external and internal cytoskeletal forces to maintain shape and integrity. So we use a technique called micromanipulation, where we can isolate a single nucleus from a living cell and hold it between two pipettes. We can create extension by pulling with one pipette, and then we watch deflection of the other force pipette. Its deflection times a pre-measured bending constant allows us to back out force, so we get a force versus extension relation, and this provides a spring constant. We have previously published in MBoC numerous occasions that chromatin dominates the short extension force response, and lamins bend but then stretch at longer extensions to create a strain stiffening effect. And here from this example force extension, you can see that all of our DNA damaging agents decrease the chromatin short extension based spring constant, but do not impact the strain stiffening that lamins cause at longer extensions. And so what actually is changing inside of the nucleus? Well, we know that the chromatin based force response regime is largely dictated by heterochromatin. So then we went in and looked at heterochromatin, specifically constitutive heterochromatin marker H3K9 dientrimethylation. And we saw that upon DNA damaging agents, we saw a drastic decrease in levels. And we hypothesized that this might be due to the fact that ATM, which is activated early in DNA damage to respond and repair DNA, is known to decompact the DNA so that the DNA can be accessed and repaired. So we used an ATM inhibitor, ATMI, and in fact, that completely rescued loss of constitutive heterochromatin. If this is true, if this is the mechanism we're working under, then we should also restore mechanics, shape, and integrity. And in fact, that's what we do. Let me walk you through the data. Here you can see our DNA damaging agent decreased the short extension chromatin-based force response. But upon treating with ATMI, we can restore mechanics in an example image. And here at the bottom across all of our measurements. And that, of course, a stronger nucleus can maintain shape and integrity. So we then suppress nuclear blebbing, shown here in DNE. And of course, blebs lead to ruptures, so we're also suppressing the ruptures that occur. Overall, what we found is that DNA damage activates ATM, which causes decreased heterochromatin, which weakens the nucleus to then cause nuclear blebbing and rupture. What's interesting is that these have a feedback between them, because we know that blebbing and rupture causes DNA damage, changes in transcription, and changes in cell cycle control. And that increased levels of DNA damage that are occurring can then feed back through the system and cause this sort of doom spiral, if you will, causing a progressive worsening of the disease state, which lines up with the fact that more severe disease states show more abnormal nuclear morphology and more nuclear blebbing. And last, I want to thank Nebiat, who did the majority of the work alongside of the large number of people we have in the lab. And here's a professional image of Nebiat, and here's a non-professional image of Nebiat partying with the lab. Thank you for your time and listening to me. Thanks.
Thank you, Andrew. Congratulations on guiding an undergraduate to produce such nice work. We might hold questions for the moment. I might then hand over to Alex Dunn to introduce the next speaker.
Yeah, sure thing. It's my great pleasure to introduce our next speaker, Ivy Han. So Ivy did this work also as an undergraduate at Vanderbilt in Andrea Page McCaw's lab. And she is now an MD student at Johns Hopkins studying oncocardiology. So as not to take away from your time, Ivy, I'll hand it over to you now.
Okay, great. Thank you so much. I'm going to share my screen. Okay. Can everyone see my screen okay? Great.
Yes.
Today I'm presenting on behalf of the Page McCaw and Hudson Labs on our work on the changes in epithelial tension after wounding and how the restoration of tension was found to be GPCR mediated. Epithelial wound healing must respond to disruptions in cortical tension in order to maintain that crucial protective barrier. So to learn more about this process, our aim was to map out changes in tension after wounding and determine modulators of those tension changes. Our experiments were done in vivo using the Drosophila puponotum, which is a monolayer epithelium. And then the pannier domain, which is shown here in orange, allows us to drive genetic manipulations only in this area and compare it to an internal control. We used a laser to make wounds at the pannier border and measure tension around the wound at different positions. If you imagine an axis going through the wound, we'll use positive positions to refer to the pannier domain and negative positions to refer to the control domain. To measure tension at each of these positions, we severed a cell border with a laser microablation and analyzed the recoil rate of the corresponding junctions. Higher recoil velocities correspond to higher cortical tension. We first looked at a spatial map of tension in the unwounded epithelium and found that tension was relatively homogeneous. We also confirmed that we were able to detect a reduction in cortical tension using organisms with pannier-driven ROCK knockdown, which reduces contractility. We then mapped out tension in wounded samples with measurements made 1 to 10 minutes post-wound. We found that there's lower tension near the wound, both radially and medial-lateral borders, and tangentially in anterior-posterior borders. Next, we used a temporal approach to assess if there are changes in tension over time, and we found that close to the wound, there was actually a restoration of tension at later times. Given this, we also wondered if there are dynamic changes farther away from the wound, and excitingly enough, when we measured tension at further locations within 30 seconds of wounding, we found that there was an initial loss of tension that we had previously missed. Thus, there's a widespread initial loss of tension around the wound that is restored over time, starting from farther away and then moving toward the wound. This made us wonder if this was related to a previously reported contractile wave that progresses in a similar manner. Here's a video showing the post-wound contractile wave visualized with non-muscle myosin II, which temporarily accumulates apically in each cell. Cell contraction is not well visualized in this video, but each cell contracts in turn after the myosin accumulates. When we measured tension before and after the wave progression, we found that tension did indeed temporally correlate with this contractile wave. We also wondered, however, whether there were other modulators of tension, specifically wound-induced calcium signaling. Our labs previously discovered that the GPCR Methuselah-like 10 is activated in epithelial cells around 45 to 75 seconds after wounding and triggers the G-alpha-Q signaling pathway, resulting in calcium release. Especially with the impact of calcium on the actin cytoskeleton and cellular contraction, we investigated tension maps in organisms with pannier-driven Methuselah-like 10 knockdown. There is no difference between the control and pannier domains in the unwounded epithelium. However, in wounded samples, there is low tension throughout the pannier domain, indicating a lack of tension restoration. We confirmed this with a temporal study and found that low tension persisted even after tension restoration should have occurred. Thus, we concluded that there was no restoration of tension around the wound in the absence of Methuselah-like 10 signaling. Interestingly, the contractile wave was not disrupted in these samples, despite methuselah-like knockdown on the pannier side, suggesting that cells may transiently increase tension and contract, but this tension is not maintained in the absence of methuselah-like calcium signaling. To summarize, we found that wounding initially results in a widespread loss of tension, which is then restored out to in, correlating with the contractile wave. The GPCR methuselah-10 is required for restoring tension, but not required for the contractile wave. So we thus propose that the contractile wave may temporarily increase tension through cellular contraction, but methuselah-10 initiated calcium signaling is required to maintain that tension after the wave progresses through. Interestingly, our labs have previously shown that methuselah-10 knockdown decreases survival after wounding. So this newly discovered tension restoration function of this GPCR may play an important role in wound healing and survival and deserves further study. And with that, I'd like to thank the members of the lab for their support and our funding sources, and thank you all so much for listening as well.
Great, thank you very much. Everybody's got questions. There are already some that have been dropped into the chat. Please keep adding to them, and we'll return to them at the end of the session.
Actually, Alex, I think we've got, oh yeah, we should probably return to them at the end of the session.
Yeah.
I'm just watching the time. But so yeah, Alpha, can we?
Okay, it's back to me now. So let me
Yeah.
introduce our next speaker, who's Irina Ivavskova from UPenn, and she's going to tell us about lipid droplets. Irina, it's all yours.
Can you see my slides and can you hear me?
We can hear you. The slides are in, you have to change to presenter mode. Perfect.
Is it already okay?
It's all good.
Okay. Thank you for giving us the opportunity to present this work, which is I'm presenting on behalf on our lab, and especially the first author, Michael Tobin, who did this work during his PhD. So this work was largely motivated by our initial studies on lipid droplets, biophysical interaction of the lipid droplets with other organelles. And we found that they have high interfacial tension, which basically makes them to behave like rigid inclusions in cell cytoplasm, and when they interact with other organelles, they can deform them and disrupt them significantly, including the actin cytoskeleton. So basically, we asked the question, what is the direct physical impact of lipid droplets on phagocytosis? Because macrophages often can be filled with lipid droplets, and they heavily rely on the actomyosin machinery to clear foreign particulates. So the system that we used, it's THP1 THP1 cells, macrophages, which are commonly used cell line. We use lactic acid to induce lipid droplets, so you can see how they are really small, very round in these images. And only after 24 hours, they're already filled with these lipid droplets, and we investigated and found that the phenotype of the macrophage is not changed towards different polarization like M1, M2, basically excluding that this could be a differentiation effect. And then we perform phagocytosis assay where we fed the cells with cancer cells, nanobits, microbits, and also retrovirus, and consistently found that there is a 30% suppression of the phagocytosis across all these different range of targets. And interestingly, although this might seems not like a very huge effect, we found in the literature that a reduced macrophage phagocytic capacity by 30% is observed in studies in aging individuals, of macrophages of aging individuals. So further to see if there's no metabolic effect of these lipid droplets, we perform other experiments where we fed the cells with micron-sized bits of similar size that are the lipid droplets, and then perform phagocytosis on 6 micron bits. And also we found a similar suppression of phagocytosis by 30%. So to go deeper to understand what is going on, we look at the actin cytoskeleton, and we perform confocal imaging, looking at the Z-stacks, and found that the redistribution of the actin in those cells is actually changing, with more actin accumulating on the base of the cells without any change of the overall mean F-actin intensity or cell projected area. So also we found that the typical drugs that are used for actin and myosin inhibitors, so tranquilin and blebbistatin, have no significantly added effect, maybe because the network is already disrupted by the lipid droplets. So further to look for a mechanism, but also to see in more realistic situation what happens with the macrophages filled with lipid droplets, which often they have to work in confined spaces, and they have low cortical tension of about 0.1 millijoules per meter, which cannot sustain the pressure in tight tissues. So we performed these experiments with micropure confinement system, and we found surprisingly that confinement actually somehow helps the phagocytic ability of the macrophage, and it's increasing by 50% where they're filled with lipid droplets. And we found that the mechanism is by the activation of the myosin here, measured by the phosphorylated myosin light chain increase in confinement, both in immunofluorescence and Western blot. And again, very surprisingly, we looked further, okay, so what happens if the cells are filled already with lipid droplets and when they're overfull? And interestingly, we found that lipid droplets abundance or bits abundance also rescues phagocytosis via actomyosin activation. So you can see this V shape where there's kind of a dip, but then almost goes to the initial capacity. And this is the good news, but the bad news is that when cells are filled with lipid droplets, they might adapt and be able to eat more when they're full, but they cannot migrate effectively. And we confirmed this with using transfer migration assay, where we saw a significant reduction in their capacity to migrate. So with this, I would like to summarize. I basically showed you that macrophages, when they're filled with lipid droplets, there's a reduced uptake because the actomyosin structure is altered, but more severe actomyosin deformation can activate myosin, and basically the mechanics behind this is by compression or stretching, we both actually kind of expand the cortical actin. And with this, I would like to thank Disher Lab, and especially Michael Tobin, the first author. This work is his baby, all the funding and all for your attention. Thank you.
Thank you, Irina. I now hand over to Alex to introduce the final speaker.
Yeah, sure enough. Our final speaker is Jordan Beach, who is an associate professor at Loyola University in Chicago in the Department of Cell and Molecular Physiology. You can see the title of his talk on the screen, so I'll let him take it from here.
Thank you, Alex, and thank the organizers and for everyone joining in. I am presenting our lab's work. I'll first introduce the people that did this work. Kihan was a really nice, great technician with us for a couple of years and has since moved back to China. was not able to present this work. Hero Patel is one of those amazing lab managers who is awesome at doing the day-to-day science, but does not want to give a webinar to all of you. And it really essential in so many of our labs. And then Stefano Sala was co-last author with me on this. He was a research assistant professor and has moved back to the UK. So you get the least and the last author presenting to you today. So, okay. So, oops. Oh, I moved out. Yeah. Okay. So, quick take home. Sometimes control experiments don't work as expected, and that's okay. I think, over the years, I've seen people give more honest presentations of how these papers are built. And this is truly one where a control experiment, went bad. And so next, we're still learning about how myosin 2 assembles in cells, and that's the second take home. I'm gonna go out of pointer real quick. Okay. So we've seen a lot of beautiful myosin 2 related mechanical forces being produced. We think a lot about how cells control the equilibrium between the monomers of actin and myosin and the polymers that are really doing this work, right? And so cells are constantly dynamically switching between a monomer state and a polymer state. And it's, of course, these bipolar filaments of myosin that we've heard in a number of talks today are producing the contractile forces that can drive these systems. And so this is just an example of that, a migrating fibroblast looking at GFP tagged myosin. You can see dynamic assembly at the leading edge and organized into these larger structures that are really pretty. But this would be true in any endothelial cell, epithelial cell, any cell we've talked about today would have really dynamic equilibrium and steady state assembly levels. If we throw this out of whack, for example, with a Rho kinase inhibitor, you can see everything basically falls apart. Or if we overstabilize the system, for example, with jasplakinolide, everything gets ripped apart, right? So we really need to be in this Goldilocks zone where cells are able to flux between a polymer and a monomer. And importantly, cells control the fraction of myosin 2 in this state in order to control overall cell contractility. And we started this project trying to get a better handle just on that, a basic question of how much myosin is in a monomer state and how much myosin 2 is in a filamentous state. And we had assays to do that over the years for decades now, really. And here's that basic assay, right? You can take a bunch of cells, hit them with a physiological Triton buffer, and centrifuge that lysate, and the supernatant would largely contain the monomer fraction, and the pellet would largely contain that filamentous fraction. And if we express GFP tagged myosin, about half would be in each of those, similar to the endogenous myosin present in those cells. And so this assay works. We could put a mutant in that would overassemble. We could show up much more in the pellet. But there's kind of two, maybe issues with this assay. Number one, Western blots are boring, right? Why do a Western blot when we can do something on a microscope? And number two, we lose a lot of resolution, right? And so we wanted to move more towards a single cell kind of imaging-based assay to start to get a better nuance of what's going on with cell morphology in certain cells, maybe expression of other players, et cetera. And so as we were doing this, we were expressing a GFP tagged myosin. You can see it in a fibroblast cell here, nicely localizes everywhere. We can hit with that same basic extraction buffer that we used here, and roughly half the signal goes away. Mostly, we think that's the monomer diffusing out of the cell, the filaments being left behind. So the basic assay works. I won't go into the details. This is, the cell morphology and aspects are parts of other projects. But the control that went wrong was the next part. And so in order to introduce that, I'm going to introduce a little bit more about how myosin monomers assemble into filaments. So the coiled tail here is really critical for building the polymers. There's a series of negative charged regions in that tail, 5 of them here, and then it terminates in a positively charged region, okay? And these positive charges are critical for building parallel and anti-parallel associations, electrostatic interactions between the monomers that then give you the bipolar filament, okay? And so this region down here in orange that I've highlighted is the assembly competence domain. And what's been shown for decades is that if you just chop that out and purify it, it can build polymer-like structures on its own. And if you delete it, then the monomer shouldn't be able to assemble into bipolar filaments at all. Okay, so this should have been our negative control. And when we first expressed it in cells, we could see really nice diffuse localization, so consistent with it being, non-assembling myosin 2. But when we hit with the extraction buffer, you can see a hint of something going on here, right? And this was our uh-oh moment. If we enhance that image and zoom in on it, you can see this is myosin 2. And this should not have been here for all the literature that we could find over decades of work, okay? So what's really nice about myosin 2, and in the past decade, the imaging advances have really enabled us to look at myosin 2 in really exciting ways, because these bipolar filaments that myosin 2 builds are about 300 nanometers in length. Okay, so we can use like structured illumination microscopy, Airy scan, SORA, etc., and zoom in on these bipolar filaments. We can express endogenous myosin 2 with a halo tag on it and look at the actual filaments in the cell and express our exogenous delta ACD and see, is it actually co-assembling with the myosin 2 that's already there in the cell using these advanced imaging techniques. Okay, and indeed, that's exactly what we see. So we're zooming in on a lamellar area here. We're zooming in on a couple of these little spots here. And these, you can see these doublets 300 nanometers apart would be a bipolar filament structure. And our delta ACD that shouldn't be assembling at all is associating really closely and right into these networks as well. So what this tells us is that the delta ACD is indeed co-assembling and building into structures that are already present within the cell. So what did we learn about myosin 2? And there's a bunch more in the paper that I won't go into for the sake of time. But we've had a series of models. And when you talk to people outside the field, they're kind of surprised that we still don't really understand how myosin 2 filaments build in cells. And so one aspect of the models is that all these negative charges here create somewhat of a cloud around the myosin 2 monomer or filament. The lights are going on here because everyone's supposed to go home, I guess. But the positive charge then should be able to help get these monomers into the filaments and through the cloud. But what we showed was that that's not the whole story, right? And that by chopping off this area, a region of the tail, and allowing it to still assemble into polymers, there must be more going on. And what we think is going on is that some of these positive charges that are already in the tail can recognize and recruit these monomers into the system. So we added, I think, a little bit of complexity in the system, but we learned a little bit from a control experiment that didn't quite work, right? So take home, one experiment's trash is another experiment's treasure, and we are still learning how myosin 2 filaments assemble in cells. They can do it without an ACD as long as there's pre-existing filaments there, and we think the ACDs that are already in those filaments are able to recruit those monomers. So I thank the key people along the way, our funding sources, and collaboration always with Patrick Oakes. So thank you.
Right.
George.
Thank you.
Alex.
Mhm.
I'm going to I realize we're out of time, but you guys are scientists and used to this kind of thing. So what I'm going to do is put the speakers back on the screen. And then, we can leave them. If the speakers could just show their cameras, that would be fine.
Mm.
And then we could see if there are questions that have not been answered. Why don't we do it this way? I'm sure that each of you have seen that there are questions written for you. Why don't we ask you to address them, if it's possible? So, because otherwise I've got to go through them all and read them all. Some of them are very difficult. Let me try.
Many of them are very long.
Yeah. Okay, so the first question was for Rong, but let's move on from Rong's. And question from Alpha Yap, and I suppose this is for Shunzi Liu. And I think you've answered the question, but could sensitivity of air-liquid interface tension vary between cell types?
Yeah, and I think that's a great question. So what we think the surface tension or the sensitivity to the air-liquid interface is really controlled by the micromorphology on the cell surface. So those are your glycocalyx, whether you have glycocalyx on top or what proteins are present on the surface. So I definitely think different cell types residing in different parts of our tissue will have a different sensitivity. And I think that's the powerful part of our measurement is regardless of the cell type, as long as you can culture them on a flat surface, we can measure the surface tension. And I think in my answer, I also talked about, I think whether this epithelial cells are adapted to the air surface or air-liquid interface or not will change their internal expression profile, and that will cause the downstream difference in the surface tension as well.
Cool. And then I asked, you mentioned that cells recover. They get peeled off that surface and then snap back as the bubble leaves. And I wondered, you say they just recover. And I
Yeah.
really wonder what that really means.
Yeah. The recovery I talked about is really a macroscopic recovery. The cell substrate adhesion at that time is at that point is completely cut off. So what we observed after the recovery. So first of all, the recovery is controlled by the actomyosin contractility. So if you use lead A, we have videos showing that they won't recover back. They will just like dangle in the fluid. And after recovery, we track the cells. I think after 12 hours, well, it depends on the cell type. After 12 hours, you start to see the cell substrate adhesion to form again. So we're not quite sure what other physiologically relevant phenomena would change, but that's the type of things we observe so far in this phenomenon.
Thank you. And then I also asked a question of Andrew Stevens, asked if cells that are typically or naturally have a lot of heterochromatin have reduced nuclear blebbing or vice versa. So I'm thinking about neutrophils or activated lymphocytes.
Yeah, I think in general, we think that if you have a lot of heterochromatin, especially constitutive heterochromatin, which is going to have this really strong concentration at the periphery, you are going to have less nuclear blebbing. It's interesting that you mentioned neutrophils because neutrophils actually have crazy shapes. And that's actually thought because they have low levels of lamin AC. And so neutrophils do away with lamin AC because it's a barrier to migration. And so they become very liquid-like and they can move quite drastically. This is shown by other groups, not by us. But it leads to this really interesting hypothesis we're working on. I think probably Dennis postulated this long before my head was able to wrap around it. But we think that actually you don't have blebs when you lose lamin AC because blebs likely need to gap lamin AC, which is strong. And so they need sort of a pressure differential or a mechanical differential, and polymer frustration can gap the lamina, and that's where you get these herniations. So if you lose lamin AC, us and many other groups have shown that you actually can't produce nuclear blebs.
You don't get a bleb, you just get an expansion of the envelope.
You just get an expansion. The edge just sort of moves in a more of a liquid fashion. You can watch these cells; they're very liquidy, much like neutrophils.
And Rong asked, how would healthy cells carry out DNA damage repair without triggering a vicious cycle of repair and blebbing?
Right. So we think healthy cells are going to be fine. We had to hammer these cells, right? We had to do a severe effect by hammering these cells with DNA damage drugs to induce tons of DNA damage. It's our belief that healthy cells going through DDR are going to be fine. But what happens as, say, you start to age or you start to slip down the progress of cancer or affliction, you're going to lose a little bit of heterochromatin, maybe a little bit of chromatin cross-linking proteins, a little bit of lamins, and that's going to make you susceptible to blebbing and rupture. Now, as you start blebbing and rupturing, as you hit this threshold level, you're going to induce more DNA damage. And then that DNA damage is going to feed back onto nuclei that are already mechanically predisposed to be problematic and can continue to drive them towards more nuclear blebbing, right? And this can be seen in something like prostate cancer, where healthy cells do bleb. They bleb at like 1 to 2%. But as you start to go down the path of a well-handled prostate cancer cell, the blebbing ratchets up to maybe 3 or 4%. But then when you get to severe late-stage prostate cancer, you start to get to 8 to 10%. So it's not like it's moving slowly. It's moving sort of more in an exponential fashion.
Great.
That's our hypothesis, at least.
Thank you. There's a question for Ivy from Alpha Yap. Is there any role for MLCK in the calcium-driven contractility that you observed?
It's a really good question. We did not measure tension after wounding with MLCK knockdown or rock knockdown or anything like that, but it's definitely possible and definitely a good idea for a future experiment.
And then Alex asked, is it known what triggers the initial contraction wave?
Also a good question. There's been previous work showing that it's likely multifactorial. Calcium can play a role, as well as other proteins involved. I think the rock can play a role too. Interestingly, we did do experiments in another kind of wounding for which we cannot see the classic contractile wave coming in, but we found that in those kinds of wounds, methodologies signaling is also required to restore tension. So the exact interplay between the two is unknown as of now.
Great, thank you. I'm trying to limit this to 2 questions per panelist. So I'm going to move on to Irina. And the question from Rong Li was, could the effect on confined migration be due to simply a change in cell size?
Yeah, that's a great question. So we didn't actually, this is also addressing partly and Alex's question. We didn't look carefully on the volume of the cells, but the area, the projected area, showed that there's no difference between loaded and unloaded lipid droplets. And again, this is a system which is very fast induced with lipid droplets filling. And yeah, the constricted migration, basically, what we found is that the lipid droplets, when they're in the pore, they further deform the nucleus. So essentially, they're adding more constriction and effectively shrinking the pore size from the perspective of the cell because they're rigid.
So this is, I'll ask Valerie's question, which was kind of related to perhaps my question, but it's about lipid composition. And oleic acid is probably changing lipid composition as well. And so does that influence your phenotype?
Actually, we don't know. We recently started to explore different lipids, not particularly in macrophages, but in other cell types. And the size and the number of the lipid droplets actually, depending on the external lipid that they were fed, it's very cell dependent. And we don't know even how to control it. I don't think actually people in the field know how to control the lipid droplet size, and definitely different lipid will have different effect.
Great.
We expect that.
Jordan, looking at these filaments,
Yeah.
how strong are they? How do you know, are they really robust filaments? I know I asked any way to measure them. I'm sure there are, but
so yeah, no, nobody's done these experiments. I mean, people have tried to, use a cantilever to pull myosin, muscle myosins out of itself. So we have some numbers on that. But for an individual bipolar filament of non-muscle or smooth muscle or anything like that, we don't know what's holding them to, like what the exact, carefully measured forces are that are holding them together. So as you put tension on the whole system, we don't know, how they're resisting that and how much force it would take to kind of rip them apart or pull monomers out of them. It's a good question.
And then the last question we'll ask here is from Shinzi Liu. And I think you're asking about normal myosins. Is that correct? Whether the high density of negative myosin or negative charges makes myosins in the bundle repel each other.
Right. So that's a really good question. So, so there's, these series of negatives. In between those are troughs of neutrality. And so you have to if you get the staggering wrong along some myosins that are moving along the tail, they would push apart and not want to be next to each other. But then they will find the right stagger. And this is what happens in muscle. This is why we have really uniform distances in muscle myosin, is because they have a really preferable stagger that they find. But anything else along the way is unfavorable and will repel. Yep.
Great. Thank you.
Mm-hmm.
So I will close the webinar here. I want to thank all of our panelists for really great talks and for staying on time with very limited time, but very concrete and excellent messages. So thank you so much. Thank you, Rong, for your keynotes. And thank you, editors, for your roles in this, both in this webinar and for doing a fantastic job of recruiting papers for this special issue. And I want to reiterate what Rong has mentioned at the end of her slides, which was we want to encourage you all to attend Cell Biology 2026 in San Diego from December 12th to 15th. Those of you in Chicago will enjoy the weather, and we look forward to seeing you in San Diego. Thank you very much. Bye-bye.
Bye-bye. Thanks.
Thanks.
Thank
Bye.
you. Thanks.
Thank you. Thanks so much.